Just to begin, in our research we're focusing mainly on the cytoskeleton of the cell. Within the cytoskeleton it is made up of three components, being the actin, microtubules, and filaments. In our research, we're mainly worried about the actin of the cytoskeleton, which is highlighted in that picture in green. The actin in the cell is responsible for many different processes, including cell motility, cell adhesions, cytokinesis, and cell shape.
The Rho family of GTPase is a signaling protein that have been shown to regulate actin reorganization in the cytoskeleton. GTPase also function as a molecular switch within a cell, where it has the ability to cycle between inactive GDP bound and active GTP bound. The changes between the inactive and active state is actually stimulated by the GAF protein. GAF protein stands for Guanine Nucleotide Exchange Factors. So how does it work? During the presence of GAF, it will initiate the release of GDP, so that GTP can come and bind to it and become activated. So overall, the Rho GAF protein has a 69 gene that code for it, and in particular the FGD family, it has a distinct genes, and most of them have not been well studied.
These FGD genes have been thought to be linked to different kinds of cancers. For example, FGD4 has been linked to the Charcot-Marie tooth disorder. It is believed that the misregulation of the GTPase signaling is what causes this link between FGD genes and cancer. This can cause overexpression of these FGD molecules, which then causes these GTPases to be turned on. So if you look at this figure here, we have our GTPase activated by our GAP and is bound to GTP. The GDP would come in and try and replace the GTP to turn it off. However, it's not able to do so. So the GDP is pushed back off, and the GTPase remains permanently on and is still stimulated by that GAF protein. So our goal when we're looking at the whole picture of things is to work on cloning these FGD1-5 molecules in order to see their expression in bacteria. In order to do this, we'll be able to get a better understanding of their function and role within the cell, and also we can understand why there are actually so many FGD molecules. So when we're speaking about cloning, we're talking about just cloning specific DNA sequences within the cell. We're not talking about whole organism cloning.
So we conducted a series of molecular cloning procedure to further look at the FGD expressions. The first, we did PCR amplify. We amplified our target sequence of FGD fragments. Second is the plasmid digestion. We cut our desired plasmid using the restrictions enzyme. The both end products for this experiment is then joined in ligation reaction using the DNA ligase, creating recombinant DNA plasmid. Recombinant DNA plasmid will be inserted in a bacterial host cell, and then replication process will take over. Replication process will create colonies. Colonies will be isolated in mini prep procedure. And moving on, the last procedure is restriction enzyme digest. We're going to cut the plasmid using the restriction enzyme, and we're going to run the gel analysis to look at the results. And in the upcoming slides, we're going to look in detail all the 6 major experiments. So first is the polymerase chain reaction, PCR. We used our FGD sequences as a target sequence. We amplified all these FGD sequences at an exponential rate. And if you look at it, all these amplified FGD strands, we have this cut site known as the restriction enzyme cut site. These cut sites are really important for the next procedure because it allows us to cut it at a specific location using the restriction enzyme.
So after this PCR amplification, we're hoping to have a lot of different copies of our certain DNA sequences. So in this case, then we'll run an agarose gel to check and make sure that we were actually able to amplify the sequences we were hoping to amplify. So this picture here shows our gel attempt for that. Here on the left side, we have our ladder. This indicates different lengths of base pairs found, so this allows us to use it as a marker to see the sequences we were hoping for. As you can see, this bottom band here indicates 500 base pairs, and then it goes up by 500 base pairs each line fragment. We'll mostly be concerned about fragments showing up between the 2,000 and 3,000 base pair range. So if we continue on and look, our first well here is for FGD1. You can see that we don't find a bright band at all, and there aren't any results showing for FGD1. So we have this black circle here to indicate where we would have hoped to see a band for FGD1 between the 2,000 and 3,000 base pair segments. However, since we were unable to find a band there, we determined that the PCR amplification did not work for FGD1. When you look at FGD2, the results are similar. Again, we have this black circle here to indicate right around 2,000 base pairs, where we would have hoped to see a band indicating a sequence. However, once again, the amplification did not occur. The results get a little bit better for FGD3 here. You can see this bright band right here. It lies right between 2,000 and 3,000 base pairs. Probably an FGD3 sequence. So we'll be able to continue on with this one in upcoming procedures. The same goes for FGD4 as well. You have this other bright band right here, also right around the same base pair length. So we'll continue on with FGD4 in our next procedures as well. When you look at FGD5, once again, you can see our black circle here, which indicates that we are unable to find a band at the sequence location that we are hoping to. Okay, so this here shows these orange bands are indicating our PCR amplified DNA. So what we need to do now is we need to cut these DNA sequences in order to provide them to be able to hook on to our plasmid. So these red sequences right here indicate a cut site for certain restriction enzymes. In this case, it corresponds with EcoR1. EcoR1 will then cut the DNA strands at this dotted line here. And once the strands are separated through a series of procedures, we'll have two different DNA strands, as you can see at the bottom here, which will both have these sticky ends indicated by the red letters, and these sticky ends provide attachment for later on procedures.
In the plasmid digestion procedure, we use our plasmid PC12 FYFP. We cut them using the restriction enzymes. So we use EcoR1 and also NOT1. After we cut them, they will have this two overhangs, they're otherwise known as the sticky ends. Moving on is the ligation reaction. In this ligation reaction, we use the same cut plasmid and then earlier cut FGD sequence. We join these two cut sequences using the DNA ligase. DNA ligase will form phosphodiester bonds between these two sequences, creating recombinant DNA plasmid. Next is the bacterial conformation procedure. In this procedure, we use the recombinant DNA plasmid. We insert them in a bacterial host cell. In a bacterial host cell, how does it work is actually the DNA and cell mix will be kept on ice and then exposed to a high temperature change. This sudden temperature change will drive in some of this recombinant DNA into the bacterial host cell, and then conformation will take place, and then there comes the replication process. During the replication process, colony will be formed, and that's a plate that we got. If you look at it, these are all the colonies. And another thing, only when we talk about these colonies, actually some of these colonies they have the recombinant DNA FGD sequence, and some of them they just plain colonies from the bacterial host cell.
So now we have all these colonies on our plates, but we want to make sure that we do indeed have the right plasmid in these colonies. So we'll go through our mini prep procedure in order to re-extract these recombinant DNA sequences from the bacterial cells. So what we essentially do is we'll take out our plasmid DNA from our bacterial cell. Once again, our restriction enzymes come back into play, and we'll want to cut out our FGD3 sequence. So these will cut at the same sites as they did previously, and this will allow us to take out our FGD3 or 4 sequence in this case, and be able to run it on another agarose gel in order to determine if, once again, we are able to get the correct number of base pairs for these sequences. So on this slide here, we have our results after our restriction enzyme digest. You can see we chose a couple colonies for each FGD3 and 4, just so that, like he had said, that not all of them will have the plasmid in them, This way we cover a more variety to try and capture our recombinant DNA. So here we have these bright bands that show up about 3,500 base pairs. This, in fact, is actually our plasmid. This is too big to be our FGD sequence. So these we can disregard because it's just showing our plasmid. If we compare this to our other gel that we did for PCR amplification, we would have expected to see our FGD3 and 4 sequences, which we could see over here, right in this general area between 2,000 and 2,000 base pairs. As you can see, we don't have any bands in this area. Therefore, we can determine that our procedure was in fact unsuccessful. So we will need to go back and start over and try and reinsert these FGD3 sequences into the bacterial cells.
So we repeated the same procedure by changing some of the experiment conditions. For example, in PCR amplification, we use a brand new cDNA form to better capture the FGD3 and 4 sequences. These arrows are indicating the band sizes for FGD3 and 4. Since they are corresponding to the right ladder size, we're able to continue with our procedures to get a better results. And then in the mini prep procedure, we picked 6 different colonies for each FGD3 and 4. If you look at an FGD3, this arrow is indicating the recovered clone FGD3 sequence. Therefore, we conclude the result for FGD3 was successful. However, for FGD4, you don't see any band sizes for the FGD clone sequences. These bands are actually for the plasmid. Therefore, we conclude the results were unsuccessful for FGD4.
Okay, so all in all, we were able to get a positive clone for our FGD3 sequence. Had we had more time to continue working on our experiment, we would have taken the sequence and sent it in to a company, and they can verify then if it's correct. This would just be a double check just in case what we're seeing is not what we really think it is. Another thing that we would try and do with our FGD3 sequence is express it in cells, and this would allow us to start determining its function. Another big thing that we'd work on is we'd go back to the beginning again and try and change some of the conditions and re-PCR amplify FGD1, 2, 4, and 5, since both times we were unable to get results for those molecules.
So over this past 2 semesters, we gained some meaningful experience by doing this research. We got to learn the ABCs of molecular cloning and all the skills, which is really useful for the graduate school. And then, apart from that, I got to know Stat. She's really, she's awesome. She's hardworking, on top of things, which I'm totally opposite of. So.
Yeah, we had a good time working together. It was different, especially coming from 2 different cultures too, so we got to know each other a lot better. Another big thing is it got really frustrating during the year. I mean, we were both, our first results, you can see we didn't have anything. So to get a gel like that, we were both pretty upset. But it's nice that we were able to finally get some results at the end, so that was exciting. And just we both did learn a lot that we can use later on in our biology careers.
Is there any questions?
Not a question, but a comment. I think you both did a beautiful job explaining the topic.